Before I begin, as a disclaimer, all of the images taken for this lecture are from the Immune System 4th edition, and they're copyrighted by Garland Science 2015. So the plan for today, I'm going to start with talking about the innate immune response. With that, we're going to talk about the beginning of complement, and we're going to talk about the induced inflammatory response, all of the adaptive immune response, B cells and T cells. We're going to talk about their activation, and then we're going to summarize with vaccines and start to finish. So the innate response happens from start point of pathogen exposure to about 4 hours, and it's comprised primarily of physical barriers, skin, mucosa, complement, asterisk, because this is the major component of this innate response. Macrophages are within every tissue within our body, and they're often the first of the leukocytes to get exposed to a pathogen. And then we have proteins that are just constitutively expressed, like defensins and alpha-2 macroglobulin, that act to kill any sort of pathogen, or at least opsonize it so it can't get into the body. So let's talk a little bit about complement. Complement is a series of proteins that are made by the liver, and they're secreted as zymogens. So they're not active in your body natively. They must be activated. And that's important because it can have autoimmune consequences. They're distributed from the blood, the lymph, and the extracellular fluid. And there are 3 stages to the activation. There's the alternative, there's the MANNOSE binding pathway, and then there's the classical pathway. So within the innate response, the one that's going to be active first is the alternative pathway. Within the blood, we have C3, complement 3, circulating throughout the blood. It can randomly be hydrolyzed to IC3, and from there, it can then interact with protein B. And protein B can bind, be activated by protein D, and create this complex of BB and C3BB. And that is the C3 convertase. And what that's going to do, it's going to convert circulating C3 into C3b and C3a. Whenever we're dealing with these, the A's become anaphylotoxins and leave into the body, and C3b generally gets embedded within a pathogen surface. Once it's embedded, it then can bind to another protein B, and again can be activated to form another C3 convertase. Once it's done that, it can then create more of the C3b, which will embed along the pathogen. This is important, is the C3b is a marker for phagocytosis by the phagocytes within the body. Additionally, the C3 convertase, again, C3b, BB, so many letters, is stabilized by propyridine, which is just a cofactor that allows this convertase to be stable within the membrane. So it's just important to know that propyridine allows it to be stable in the membrane. And then we have several mechanisms by which we can inactivate this system. The first of these is the HI system. There's protein H and protein I that both can bind to this embedded C3b and act to cleave it and turn it into IC3b. IC3b can no longer bind and make a convertase. However, IC3b can bind with CR1 and CR3, which are the complement receptors within the body, which is when those bind, it's a signal for phagocytes to take up that pathogen. So you get a pathogen that's coded with C3b and IC3b, both of which signal for phagocytosis. There's a couple other mechanisms by which we can suppress this. We've got proteins DAF, decay accelerating factor, and MCP. Both of these act to prevent the formation of a convertase, so you don't continue making the C3b, and also serve to generate IC3b so that, again, it stops that pathway of the convertase. As the final step in the complement pathway, a convertase can convert C5 into C5b, which can then combine with C7, C6, C8, and C9 to make this pore in the pathogen surface, which is called the membrane attack complex. Essentially, once this pore is made, it allows the interstitial fluid to flow within, continually with the cytoplasm and kills the cell. This is an essential part of our innate immune system because that membrane attack complex is going to be the first thing that is going to start killing the bacteria before any of our immune cells are activated. We also, as I mentioned, have macrophages. Macrophages are a phagocyte, and they have a bunch of receptors on their cell surface. Of note, none of these receptors are ever variable. They're encoded in your genome, and their goal is to detect wide classifications of proteins that wouldn't be native to the human body, and then phagocytose them. So if you have, for instance, here it displays a bacteria and a yeast. They have surface proteins that aren't normally within our body, so the macrophage will recognize them, absorb them, and then combine them with a lysosome to make a phagolysosome. I wanted to point out, because it was mentioned in class, one of these receptors is the TLR4 receptor. The TLR4 receptor is really important because it recognizes liposaccharide or lipopolysaccharide, which is very common within bacteria. Additionally, I wanted to point out that CD14 is the receptor that initially recognizes the lipopolysaccharide, and you require this MD2 protein in order for TLR4 to bind. Without the MD2 and without CD14, TLR4 would not recognize that lipopolysaccharide. So let's talk a little bit about TLRs. TLRs are toll-like receptors. The main goal of a toll-like receptor is to induce inflammation. So a signal from a toll receptor almost always ends in the activation of NF-kappa B, which is a transcription factor that induces inflammatory cytokine production. There's some of them that are, I think, high yield of note. Again, TLR4 with the lipopolysaccharide. And this is going to be responsible for systemic shock very frequently. We have two classifications of TLRs, those that are on the surface and those that are endosomal. Endosomal, as one would imagine, are going to recognize things that get phagocytosed and are inside of an endosome. So that requires the bacteria to have been broken down. So you're going to get exposure of things like RNA and DNA that the TLRs that are in endosomes are going to be able to recognize. And meanwhile, surface TLRs are going to detect all sorts of surface markers that are on pathogens or parasites. So I'm going to transition and talk about the induced inflammatory response. This happens between 4 hours and about 4 days. And this is the continuation of the innate immune response. It's driven primarily by macrophages. So macrophages will bind to the toll-like receptors, which will activate NF-kappa B, as I mentioned, and this will produce a bunch of inflammatory proteins. And so I've included the really important ones here because it'll start to connect the dots as we continue moving. So importantly, both IL-1 and TNF-alpha are going to cause vasoactivity and allow greater permeability. IL-6 is the liver-activating interleukin. That's going to produce C-reactive protein, fibrinogen, and mannose binding lectin, there we go, which are going to be important later in more complement activity. IL-12 is produced, which recruits the NK cells and promotes macrophage survival, as well as Th1 differentiation. Again, all very inflammatory to try and recruit proteins, recruit cells. And then CXCL8, which is a neutrophil chemokine, so it's going to try and recruit neutrophils. And then, as I mentioned, we have anaphytoxins. These are the C3a and C5a. And these are the byproducts of activating complement, but they are also activating themselves for mast cells, basophils. And what this is going to do is it's going to release histamine and leukotrienes, and that's going to add to the inflammation by causing more vasoreactivity and permeability. So to demonstrate what's going to happen with neutrophils, you're going to get neutrophils that are normally flowing within the blood. However, when you get an inflammatory response, the neutrophils are going to move towards the endothelial side, and selectins will allow them to roll along the endothelium. And they're going to be attracted by this chemokine CXCL8. And again, that's released by macrophages to draw the neutrophils into the site of inflammation. Importantly here, as part of the inflammatory response, ICAM1 is triggered to be released by endothelial cells near the inflammatory source. What this does, it allows the neutrophil to follow the chemokine until it interacts with this ICAM1, at which point it moves through the blood vessel and into the site of inflammation. This is an example. The neutrophil, like macrophages, is coated with proteins that recognize foreign compounds. So they have CD14, they have N-formyl methionine receptor, which recognizes any bacterial-made protein, along with complement 4 and complement 3 receptors, which I mentioned are signals for phagocytosis. So the neutrophil moves into the site of inflammation. It is going to phagocytose any sort of pathogen, combine it with a lysosome, and break it down, much like the macrophage. The exception here is that neutrophils will commit apoptosis once they're done. So this is why we get pus, because neutrophils will commit apoptosis as soon as they're done with their reaction. And this is an innate response that is triggered at preventing systemic inflammation, essentially. So let's talk about complement some more. As I mentioned, IL-6 is going to trigger the liver to release more complement-inducing proteins, specifically mannose binding lectin. Mannose binding lectin allows for C4, in this case, to be activated to C4b, which then is going to combine with, this is incorrect according to lecture, this is supposed to be C2b. Anyway, it's going to combine, and this combination of C2b and C4b is again a C3 convertase, but this is through what's considered the mannose binding lectin pathway. So we've all circled back around to this complement system. Again, you're going to use this to activate the membrane attack complex and kill the pathogen. And then the classical pathway is just an adaptation of this, where you replace mannose binding lectin with C-reactive protein, or as we'll talk about later, antibodies. With a viral infection, you release interferons. These are a response to, again, receptors within the cell that detect viral, usually viral DNA or proteins, and they're going to induce natural killer cells, which are going to come to the tissue and kill any healthy cell, or any virus-infected cell. The reason it's going to do this by recognizing MYC protein, which we'll talk about a little bit later when we talk about MHC. But the purpose of the natural killer cell is to remove virus-infected cells from the system. All right, let's transition and talk a little bit about B cell development, because this is going to be one major step in our adaptive immune response. So before we can talk about the cells themselves, we need to talk about antibodies. This is the general shape of an antibody. What's unique about the antibodies in our system is that they can bind to many epitopes. That can be proteins, it can be polysaccharides, or really anything that can interact with their highly variable region here at the end. That makes them useful for detecting any sort of foreign molecule within our body. Importantly, they have two chains, the heavy chain here in green and the light chain in yellow. And within these, they have two regions, a variable region in red and a constant region in blue. The variable region is where you're going to get epitope binding, and the constant region, otherwise known as the FC region, is where you're going to have receptors on all sorts of phagocytes that recognize an antibody has been bound to some sort of pathogen. There are 5 classes of antibodies: IgG, IgM, D, A, and E. What's important is within the body, IgM and IgD are the default antibody that is made, and you must undergo class switching in order to express the other ones. We're going to talk about that. On the antibody, as I mentioned, there's the antigen binding site here at the end of the, this is called the FAB segment. So if you see that, FAB means the antibody binding region. And what's of note here is there's sites on this, the CDR regions, the complementarity determining regions, that are highly mutagenic. In other words, they have a lot of variability that allows them to bind to all sorts of epitopes. It's important because we want our antibodies to recognize anything that's foreign in our body. So how does this happen? Well, we have a large assortment of genes within our genome that code for all of the different components of the antibody. We have heavy chain loci, and then we have two light chain loci, the kappa and the lambda. Each one of these regions is a section of, is a coding fragment that at the end will get combined to make a functional antibody. There's, importantly in the heavy chain, we have three regions. We have the V region, the D region, and the J region, whereas in the light chain we only have the V region and the J region. So there's a little more diversity within the heavy chains. So what's going to happen is our body will pick one of the V regions, one of the J regions, and then a constant region for the light chain. And it'll pick one of the V regions, one of the D regions, and one of the J regions with an assortment of the constant regions for our heavy chain. And again, the reason we have so many more of the constant regions is because in the heavy chain you have 1, 2, 3, and 4 regions that are being made. It's a larger molecule, whereas the light chain only has the single region here that's being made. The way this is selected is through two proteins, the REG proteins. They're going to come in and they're going to excise all of the DNA that would be between the two selected regions. So here this is demonstrating you have, in a light chain, you have multiple V regions. You have V1 and V2. The REG protein is going to come in and it's going to remove the V2 so that it combines the J region with the V1 region. So you get this final protein of V1 right next to J. So the REG essentially is going to just clip out intervening pieces of DNA. I also want to note there's a 23-12 rule that was mentioned in class. This is important because the REGs don't just cleave anywhere. They cleave at very specific sites so that you maintain a functional antibody. And those sites happen to be these 23 and 12 base pair regions. So that's the 23-12 rule. Sorry it's a little bit small, but there's this additional protein, TDT, which adds more variability to the antibody by inducing random insertions of nucleotides. Oh, my apologies. Wow, I have no idea what's happening. I'm so sorry. My computer just freaked out a little bit. There we go. So we have random insertions of nucleotides between those segments that are being combined by the REG protein. Random bits of nucleotides are inserted there to add variability. Again, all of this is designed so that the antibody can have as much variation and bind to as many things as possible. So that's the TDT protein, just adds variability to it. So there's an additional component which is called class switching. This is how we get all of our 5 different antibodies. They have to undergo class switching. This is done by the AID protein. What that does is it adds nicks into the genome between sections of the constant region. So to illustrate this, if we imagine we have a gene fragment that looks like this with multiple constant regions, what can happen is the AID protein comes in, clips out a section of it, and then it's recombined. The reason this is essential is because when an antibody is being produced, the formation is always going to just go in this direction. So that what happens is you always code for the constant region that's closest to your variable region. So if we look at our gene sequence again, we have multiple constant regions here that are closer to the VDJ region, And these would be coded for first, namely this mu protein. This is why our IgM is the default antibody, because on the genome, it's the closest to the variable regions. Does that make sense? All right, so how is this all done? What's the timing of the whole thing? Well, this all happens within the bone marrow, with the exception of class switching. We'll talk about that later. But I wanted to point out that a B cell receptor is an antibody attached to these coproteins. It's important because the B cell receptor is the antibody that would be produced by that B cell when it's mature. So whenever we talk about B cell receptors, we're talking about bound antibodies, essentially. So in the bone marrow, we have stromal cells. These are important because they are providing the survival signals for the B cells to mature. And so the B cells are going to come in and they're going to bind through their entire development along this stromal cell, which will again provide them the survival signals to continue their development. So the first step in B cell development is the recombination of the heavy chain. The heavy chain is made first. You start by combining the DJ regions, and then you add the V region to it. That I don't think is important. What's important, though, is that the heavy chain is made, and then there's a checkpoint in which we have a surrogate light chain. This is default in all B cells. Surrogate light chain comes in, binds with the heavy chain that's been made, and that's checkpoint 1. The B cell must have a functional heavy chain or it will perform apoptosis. So that's checkpoint 1. From there, the light chain is made. If it passes the checkpoint with the surrogate light chain, it then undergoes the somatic rearrangement to generate a light chain. Kappa and lambda chains are made sequentially until a functional antibody, or in this case a B cell receptor, is made. So if the first time it undergoes the somatic recombination, if it makes a functional kappa chain, it'll just continue and that will be the B cell receptor. If, however, the first chain does not result in a functional B cell receptor, it then moves to the next one and it tries the other chromosome's kappa chain. If that doesn't work, it moves to the lambda chains. So you have 4 chances to make a functional light chain within the B cell. From there, it goes to the third step, which is selection. It goes through negative selection, in which the proteins in the bone marrow, or the cells in the bone marrow, are going to partner with these developing B cells and present self-proteins. If the B cell binds too tightly to a self-protein, that's a bad thing because you don't want antibodies against your own cells. So what the B cell is going to do is it's going to have 2 chances at essentially regenerating itself. So if it binds and it's self-reactive, the light chain undergoes another set of rearrangements. Remember, there's 4 chances with the light chain, so you've got extra sets. It's going to undergo more mutation in the light chain in an attempt to make a non-self-reactive protein. So if the B cell receptor is still self-reactive after that second attempt, it then commits apoptosis. And when the B cells have finally made a mature receptor, they're not self-reactive, they get released and they are considered naive B cells. If they come out of the bone marrow and they're not activated, they are naive cells, and that goes with T cells as well. Of note, all B cells express CD20, so if you're trying to detect B cells, CD20 is the marker for that. And as I mentioned, class switching has not occurred. So our 2 B cell receptors we're making at this point are IgM and IgD. All right, let's transition to talk T cell receptors now. Excuse me, T cell development. So this starts with MHC. MHC is a protein that's expressed constitutively in every nucleated cell, which would not be red blood cells. And we have 2 types. We have MHC1 and MHC2. They each bind to a constitutive T cell receptor, or co-receptor rather. So MHC1 binds to CD8, MHC2 binds to CD4. Important, and this is essential, only peptides can be presented on an MHC molecule. They are also degenerate, which is a term that means that multiple peptides can bind to the same MHC molecule. MHC1 is the one that's present on all nucleated cells, while MHC2 is limited to antigen presenting cells. We have a total of 6 MHC proteins, 3 from each parent, and that goes for MHC1 as well as MHC2. When we're talking about an HLA subtype, we're talking about a type of MHC. So those are used fairly interchangeably, just to clarify anything. Subtype A through C are the immune presentation ones. And again, we have 3 of those, A, B, and C from our mother, and then 3 A, B, and C from our father. HLA-G subtype is the subtype responsible for fetal-maternal interaction. And HLA-E is a subtype that's used to detect healthy cells from virally infected cells. This comes back to the natural killer cell attachment and recognition. So MHC1 is a molecule composed of 3 chains connected, alpha 1, 2, and 3, with, this is beta 2 macroglobulin. It has notably closed ends, so that the fragments that bind can only be 8 to 10 peptides long. It's important because MHC1 is limited in the peptides it can bind. MHC2, however, has open ends, and as a result, it can bind longer peptide chains that can have a little more variability. As I mentioned, the receptors are degenerate so that multiple peptides can bind to one receptor. And then as a way of recognizing, sometimes you may just see a picture of an MHC interacting with a T cell, and it might not give you details of what type of MHC and what type of T cell. So I just remember that MHC2 has 2 anchors in the membrane. Always clarifies whether it's dealing with a CD4 cell or a CD8 cell. If we're talking about MHC1, this is made within the endoplasmic reticulum, and it's actually going to have the peptide fragments loaded in the ER. And so there's a protein TAP, which transports small peptide fragments into the ER, where they get loaded onto the MHC1 protein and then sent out via the Golgi body to be expressed on the cell surface. Of note, the proteasome actually can be converted to an immunoproteasome when inflammatory cytokines are present. So for example, interferon gamma can induce the immunoproteasome. It's important because the immunoproteasome generates fragments that are 8 to 10 peptides long, which is important because MHC1 can only bind fragments that are 8 to 10 peptides long. Additionally, MHC1 is particular about it must have a carboxyl end and an amide end, and so this immunoproteasome is just making fragments that fit into MHC1. And of note, I'm not going to go over all of them now, but viruses make proteins that block this and prevent this presentation. So this is how viruses stay within our cells and are able to evade immunodetection, because if they block any part of this process, MHC1 is not going to express itself and is not going to present the viral peptides. MHC2 has a different pathway for getting loaded. This is done within endosomes themselves. So within the ER, this invariant chain is placed as a placeholder within MHC2. It's loaded into vesicles. When those vesicles are combined with an endosome, these combine, all of the fragments are going to then be able to be loaded into this site that again has a placeholder now called CLIP. So CLIP gets pulled out by this HLA-DM protein and is replaced by one of the fragments from the endocytotic vesicle. Does that make sense? This is how MHC2 allows for presentation of exogenous pathogens. MHC2 allows exogenous pathogens to be expressed. MHC1 has to have things that were already in the cell because it had to have gone through a proteasome. Make sense? This is again why APCs, antigen-presenting cells, are the ones that have MHC2 because they're going to be phagocytosing things from the environment. Normal cells aren't going to be doing that. There was also a concept that was mentioned called gene conversion. This is a process by which the MHC molecules are always increasing their variability. This happens within mitosis, not meiosis. I asked Dr. Gleipter about it, and in the cell it says it happens during meiosis. In fact, it happens during mitosis. As the oocytes are mitotically dividing, this gene conversion takes place that essentially shuffles the genetic material so that there's always a new generation of more variable MHC proteins, which is important because our MHC determines what we're able to present to our immune system. So you can imagine if you have an MHC molecule that doesn't bind to cytomegalovirus proteins, for example, you won't present it, and our immune system won't detect it. So it's important to keep the variability within the population. So let's talk about these cells themselves. Let's talk about the T-cell receptor. T-cell receptor is essentially just the antibody fragment of an antibody. So you just take this end fragment, stick it in a membrane, and that's essentially a T-cell receptor. They have these coproteins, CD3, it's expressed on all T cells, and it stabilizes the receptor. Of note, they have an alpha chain and a beta chain. These are kind of like the light chain and the heavy chain from the antibody generation. They are MHC restricted, which means that they have to pair with an MHC molecule. A T-cell receptor is worthless if MHC isn't around. And as I mentioned, they're limited to a peptide. And this is because MHC is limited to a peptide. So if a T-cell receptor can only see MHC molecules, then only things that are attached to MHC can be seen by a T-cell. There are co-receptors, so there's CD8 in CD8 T cells. These are going to interact with MHC class 1, and this reaction will induce the cytotoxic T cell. And there's CD4, which is going to interact with MHC class 2. Just note there are two types, CD8 and CD4, and that each of them correspond to a different type of T cell, where CD4 T cells are helper T cells, while CD8 T cells are the cytotoxic T cells. There's also a bunch of genetic variability within T cell receptors. And this is very analogous to the antibody gene recombination. So the T cell receptors, you pick one of the V regions, you combine it with a series of J regions and a constant region to get your alpha chain. Likewise, you combine a series of V regions, D regions, and J regions to get your beta chain. So let me see, I can't remember if there we go. So you can think of this, which is how I do it, as the light chain is the equivalent of the alpha chain in the antibody. They each have less genetic regions, and the heavy chain is the corollary to the beta chain. Does that make sense? So T cells begin in the bone marrow and then are moved to the thymus. In the thymus, they are called educated, where they essentially are presented all sorts of self-antigens and selected for those T cells that aren't self-reactive. That's education. So in the thymus, there's several regions. There's a cortical region and a medullary region. The cortical region is where the thymocytes are going to enter. They're then going to proceed upwards, where they're going to interact with cortical epithelial cells. At this point, the cortical epithelial cell is going to present MHC1 and MHC2. And the T cell is going to interact with the MHC molecules. If a T cell is able to interact with MHC, it's kept. This is called positive selection. If, however, the T cell does not interact with an MHC, it's killed and performs apoptosis. It's important because, again, T cell receptors can only interact with MHC. Yeah.
At that point, is it interacting with both MHC1 and MHC2?
Either or. It doesn't matter which one it interacts with, it just has to interact with an MHC molecule.
But is it expressing both CD4 and CD8?
It is. At this point, it's a double positive cell, which gives it the ability to interact with either an MHC1 or an MHC2.
But it's not going to interact with both?
It doesn't have to. It could, but it doesn't have to. Does that make sense? And again, this is all based on this random chance of that genetic variation, because each one of those gene combinations makes a different receptor, which has the potential of binding to MHC or not. So this is why we have so much genetic variation and this positive selection to make sure that you are able to interact with MHC. So here this denotes the positive selection. If it binds to an MHC molecule, it is then sent onto the medullary region and it's kept alive. If it doesn't bind, it performs apoptosis. It then proceeds to the second step of T cell development, which is called negative selection. Just like B cells, you do not want to have any cells in your body that are autoreactive, because then your immune system would be attacking yourself. So we do this in the thymus through negative selection. This happens in the medullary region in here, and it's going to interact with dendritic cells and more epithelial cells that are going to present only self-antigens. This is important. The thymus does not get any foreign antigens. It only presents self-antigen. So if you ever see an interaction, so here all of these red, that's a protein fragment, but it's a self-protein. So this is why in negative selection, if a T cell binds to an MHC plus an antigen, in this case a self-antigen, it's killed, because you do not want your T cells to bind to it. Ideally, your T cell would come in, it'd be like, nah, I'm not interested, and it would just leave. And those T cells are kept. Yeah?
So even in positive selection, it's still binding to a self-antigen on that MHC?
In positive selection, it does not even need an antigen. In positive selection, it's just checking for MHC interaction.
Okay.
In negative selection, it's looking at self-antigens. Good question. Any other questions on this? I know it's sometimes a little confusing. Yeah?
At this point, during negative selection, I guess my question is just when the transition from double positive to single positive.
Good question. So in negative selection, this is the step in which it's primarily concerned with too much interaction. Positive selection, the interaction in positive selection is looking at whether it interacts with an MHC. So it's actually in positive selection that decision of whether it's going to be MHC1 or a CD4 or a CD8 cell will start to be made. And again, it's going to be all controlled by cytokine release by those epithelial cells and dendritic cells. Does that make sense? If the T cell interacts with an MHC class 1, it will upregulate CD8 and downregulate CD4. If, however, in positive selection, it interacts with with an MHC class 2, then that cell will upregulate CD4 and downregulate CD8.
So it's only going to interact with either MHC1 or MHC2, even though it's expressing both CD4 and CD8 at that point.
Exactly.
It's only going to interact with.
In theory, yes. I don't want to say only because I'm not 100% sure, but in theory, they should interact with one of them, and that would become the primary. If it interacted with both, it would probably just go with one of them, I would guess.
If you don't have any foreign peptides in the thymus, what is it for positive selection? Like, what peptide is it reacting to?
My understanding is positive selection would be, I believe, I would need to check and read on this, and I don't actually know if it says in our immuno textbook, but it is a designated self-antigen that it's used in the thymus just for the interaction of MHC. And if I remember right, it's a protein that interacts with all T cell receptors. It's one of those universal binding agents that allows for selection of just MHC interaction. But I would need to read that specifically. Does anyone else know? I'm pretty sure it's just a universal antigen that binds, that allows just selection for that MHC molecule. As I mentioned, it goes through negative selection, where any self-reactive T cells are removed. And depending on whether or not it interacts with MHC1 or MHC2 will determine whether or not it expresses CD4 or CD8. So it's, again, just all based on what it interacts with. And that's because that cell-cell interaction induces downstream signaling. And the downstream signaling upregulates transcription factors. So once they've gone through negative selection, they exit the thymus, and they are now naive T cells circulating throughout the body. Okay, now we're going to get to the activation part. We're going to start with T cell activation, then we'll proceed to B cells. Yeah?
Are they considered mature at this point? Mature naive?
Yes, mature naive B cells at this point. I believe immature is considered uneducated, if I remember correctly.
Uneducated like positive or negative?
Yeah.
So that's different than B cells where they don't become mature until they actually meet their antigen?
Yes, I believe.
So T cells don't have to meet their antigen or like do anything to be mature?
I think they just need to express the T cell receptor. Again, I would need to check on that. I think the important part is naive. Because naive implies that they're not activated, but that they could be. Right? Whereas a mature cell doesn't tell you if it's an effector or a naive cell. Right? Mature just means it's reached the end of its development. So I think that the active term would be naive, because that implies they're not yet activated to actually go in and perform their antibody production or cytotoxic activity. Make sense? All right, let's talk about T cell activation. T cell activation requires two signals, and it takes place with an antigen presenting cell. So, a antigen presenting cell, when activated, will express B7 and will express an MHC class 2. Of note, these don't have to come from the same source. Activation usually happens by TLR'S or NOD receptors or any sort of receptor that detects a foreign antigen, right? That's how you get activation of a, for example, macrophage or dendritic cell. However, everything within that cell is going to get presented on MHC. So it's important to recognize that the activation of the presenting cell does not have to be the same thing that is being presented on the MHC molecule, because everything within that cell will be presented on MHC class 2. Does that make sense? I see confused faces. So if you have a bacteria in your cell and it's producing a peptide on its surface, that peptide could activate the dendritic cell. You with me? If you have another thing within your system, let's say now a different bacteria, that bacteria will have proteins in them, right? This dendritic cell is going to be phagocytosing both the red and the green. As it does that, it could get green material loaded into its MHC class 2, though it was activated by this blue peptide. Does that make sense? They don't have to be the same thing, because the phagocyte is taking up everything within its environment and presenting all of that. So it could be the same thing, but it doesn't have to be. That's important when we talk about vaccine making and we talk about autoimmunity. So I want to make sure, does that make sense to everybody? Yeah.
So is everything presented on the MHC class 2 that was like in the cell if it gets activated?
Anything that was in an endosome. Remember, MHC class 2 is loaded in the endosomes. So anything that was phagocytosed, broken down, and is a peptide that fits into MHC2 is presented. So that could be the red bacteria, or it could be any other bacteria that's in the body. Does that make sense? Okay. The correlate of that is the T cell. The T cell is going to interact with that MHC class 2, and expresses CD28. CD28 is the co-receptor that provides signal 2 to the T cell. And all T cells use that CD28. So if it's a B7 and CD28 interaction, you're activating a T cell. Okay? And of note, this CD28 could be replaced with CTLA4. If that happens, it actually inhibits. It doesn't get that second signal, and it inhibits the T cell activation. Additionally, there's a PD-1 receptor that was mentioned by Dr. Tiwari, which if that is expressed, so if we have PD-1 expressed and it interacts, it induces the T cell to perform apoptosis. So it actually causes the T cell to die if it interacts with a PD-1 ligand. Okay? And then I have note here that a CD8 T cell, because they're cytotoxic, can often require multiple signals, more than just the 2 hits. They can also require T helper cell interactions or a third activating signal. So just know that CD8 T cells are a little bit more complicated to get active because you won't want them just running amok. Any questions on T cell activation? Yeah.
Like, I guess, what would be the reason for inducing apoptosis of a T cell? Like, if you have PD-1 being expressed and binding to the ligand?
So that's a good question. After an immune response is done, you don't want immune cells just floating around. So if you want to cut down on the immune cells that are in the system, because they exponentially increase, right? If you want to bring those down, you have to somehow get rid of them. And you signal that often with PDL. All right, I wanted to point out that the activation of a T cell uses a lot of downstream proteins, namely ZAP70, is going to get activated. It's a tyrosine kinase. It's going to go in and have multiple effects. Of those effects, the important ones to know are NF-kappa B, again, our common inflammatory pathway, AP1, which is another transcription factor pathway, and NFAT, another transcription factor pathway. So know that if your T cells are activated, they're going to use ZAP70 to activate NF-kappa B, AP1, and NFAT. And all of those are transcription factors that activate the T cell, because you need proteins to be an activated T cell. Any questions on that? All right. There are multiple types of T cells, but I wanted to point out that all of them express CD3. That's that co-receptor that goes with the T cell receptor. And they each have different functions. So Th1 is going to interact with macrophages. Th17 is going to interact with neutrophils. Th2 is going to interact with B cells. And follicular helper cells, the TFH, are going to primarily be interacting in the lymph node and the germinal centers to activate B cells. But we'll talk about that when we talk about B cell activation. And then finally, we have regulatory T cells, which are essentially going to suppress the action of other T cells. I wanted to point out regulatory T cells. They're going to use the transcription factor FOXP3. Oh, yeah, no.
Sorry, can you go back one second?
Oh, of course.
So I emailed Dr. Luter about it, and actually the characteristic cytokines for Th1 is wrong on that graph. It's supposed to be IL-2, not IL-12.
Oh, yeah, sorry.
He agreed, but never really clarified anything beyond that, just so everyone.
I also will note IL-2 is required for T cell development. So secreting IL-2 allows for T cell expansion in the bone marrow. If you don't have IL-2, you won't get that big proliferation of T cells. So thank you for pointing that out. All right, T-REGS. There's actually something wrong with this image. Does anyone have any idea what's wrong with this image?
T-REG also needs CD25 expressed.
CD25. Actually, the T-REG is going to use CTLA4. And that's going to interact with the B7 protein. So T-REGs, you're wanting to turn down the immune system, so they use that checkpoint inhibitor, the CTLA4. Whereas this one over here has the CD28. Does that make sense? So it's just, I wanted to point out that that image doesn't show the full 2-hit hypothesis. It only shows the B cell, the receptor interaction. But you indeed need those co-receptors to do any of this. Let's talk a little bit about anergy. This has everything to do with that two-hit hypothesis. The first one is it needs to interact with an MHC, and the second one is it needs its co-receptor. If the T cell interacts with an MHC alone, it becomes anergic, and it does not then proceed to get activated, even if later it interacts with its co-receptor. So that's anergy, and that's a form of peripheral tolerance. And again, it's important because let's say, for example, that a dendritic cell, this one up here, is presenting a self-peptide. If it's presenting a self-peptide, remember I said that MHC2 presents anything that is phagocytosed? So if it happens to phagocytose a self-protein and presents it, you don't want T cells getting activated. Since self-proteins won't activate the dendritic cell, it won't express that co-receptor. Because it doesn't express that co-receptor, a T cell will not be activated by interacting with it. Does that make sense? That's how we get tolerance. You can display a self-peptide, but you won't activate a T cell because that dendritic cell wasn't activated. Make sense? This is also how we get autoimmunity. Because if as I mentioned, that T cell or that dendritic cell can be activated by anything in the environment. So if it gets activated by, for example, spare LPS floating around, then it can be activated, display a cofactor, and be displaying self-peptide, which would activate a self-reactive T cell. Does that make sense? Okay. Any questions on that? Yeah. So as I spoke about, a antigen-presenting cell can be activated by anything that is foreign, right? So if you happen to have a foreign particle, a little tiny bacterial peptide, let's say, floating around in your interstitium, that can cause your antigen-presenting cell to be activated, right? If that antigen-presenting cell is activated and it just happens to be presenting self-peptide, if a T cell receptor comes in and binds to it, the T cell gets activated. Now, normally, negative selection would prevent that from happening, right? You shouldn't have self-reactive T cell receptors, but occasionally one gets out. So this is the theory of how we get autoimmunity. That make sense? Does that clarify? Summarize what I said, that they're independent activation, and they're activated by cytokines and innate receptors. So again, dendritic cells are activated independently of the T cells and independently of what they're necessarily presenting. All right, with the cytotoxic T cells, they get activated again by MHC class 1. They also use this B7 CD28 interaction. When that 2 hits is present, they then undergo proliferation and are then activated where they no longer need that second signal. So an activated T cell only needs to interact with the T cell receptor and the MHC. It doesn't need that co-receptor, which is why they take a little bit more to get activated, because once they're activated, they interact with anything, they kill it. Make sense? All right. I mentioned the Th1 cells are macrophage interacting cells. The reason I wanted to point this out is because CD40 is going to be the ligand that activates macrophages and also activates B cells. So keep CD40 in your head for macrophages and B cells, and keep B7 in your head for T cell activation. Okay? What's important is macrophages will take up all sorts of things in their environment, but often won't just default combine that with a lysosome. They need activation to do that. So you can activate a macrophage with T helper cells and the cytokines they create. Make sense? And that CD40 ligand interaction is what's going to induce that. All right. Now is the more complicated one, but personally my favorite one. This is the B cell activation. This happens in our lymph. Oh yeah, sorry, Pam.
Sorry, trying to move back to the T cells, I was just curious, when you're talking about like a foreign peptide moving around and activating an APC, I was just wondering what the mechanism of that was. Like, I'm just curious as to
So-
how exactly does that happen where it's not like being phagocytosis, like how's that just going to activate that?
Yeah, so that's a good question. So the question was, how does a macrophage just get activated by things floating around? And that comes down to those inherent receptors that macrophages have. Receptors against like a TLR receptor or a NOD receptor, or the multitude of receptors that a macrophage has against foreign molecules. So if any of those receptors interact, that's going to send a downstream signal via the many signaling pathways in the cell to activate them. By activating, it just means turning on more gene transcription. Any other questions before I go to B cell activation? Okay. So B cell activation takes place in the lymph node. And we got to do a little bit of background before we talk about the activation, because I think there's important steps we need to have before we can see the full picture. First of these is that CR2, complement receptor 2, binds to C3d. The way this is done, the C3b we talked about, it's made by all complement activation, interacts with CR1. That interaction allows for factor I to come in, cleave IC3b into C3d. That C3d can then interact with CR2. Okay, so CR1, CR3, and CR4 all interact with C3b. But CR2 interacts with C3d, which requires that additional cleavage. Okay? A little bit more background. We have follicular dendritic cells in our lymph nodes. These reside near the germinal centers, so they're going to reside in that region there. And the way that these function, as I mentioned, there's that CR2 receptor. The follicular dendritic cells use the CR2 receptor to essentially hold any sort of pathogen on their surface. That's all their function is. They just hold a pathogen on their surface. So within your lymph, you have all sorts of antigens that are combined with C3d. They enter your lymph node. They interact with a macrophage, which essentially just takes it in and transfers it to the follicular dendritic cell. Once it's there, it's presented and ready for B cells to come in and take a look. Yeah?
So when, on the last slide, when you CR2 binds to C3d, does that activate the B cell?
That does not activate the B cell. We're going to talk about it. We're going to talk about it. Because I think this is perhaps one of the most confusing parts of the lecture series, at least for me personally. So we're going to really talk about how B cells get activated. All right. So the follicular dendritic cells are just important because they are going to present all things that the B cells will interact with. Okay? So let's talk about B cell activation. B cells start by migrating to the lymph node, and they do this using CCR7, which is a receptor for CCL19 and CCL21. Both of these proteins are chemoattractants to the HEV. Okay? They're made by the stromal cells and made by the dendritic cells. Additionally, B cells have a receptor, CCR5, which interacts with CXCL13. CXCL13 is a chemoattractant made by the follicular dendritic cells. So the point of this is that you want a B cell to get to the lymph node, and then you want it to go to the dendritic cell because that's where antigens are presented. Okay? But before we get there, there's an important thing that happens. It's important to recognize that we have a T cell region, and that's denoted here in blue. So before B cell activation can happen, T cells must enter the lymph node, and in this region, they're going to interact with dendritic cells. So just like follicular dendritic cells are presenting antigen to B cells, dendritic cells also migrate into the lymph node and present antigen to T cells. At this point, they are naive T cells. Okay? T cells will come in using those same chemokines that the B cells had, the CCR7. they will come in, interact with a dendritic cell where that activation we just talked about can take place, okay? So it's important to recognize that a T cell also gets activated in the lymph node. Okay, so now that we know that T cell is activated, it's ready to go. And this is a helper T cell, a CD4 T cell, just to clarify that. So now we've got a T cell sitting and ready to go. So now let's look at our B cells and what happens with the B cell. B cells, as I mentioned, use CCL13 or CXCL13 to move to the follicular dendritic cell. At the follicular dendritic cell, there are a bunch of pathogens that are just being presented, right? The B cell, with its B cell receptor, can interact with those antigens. That interaction activates a cascade of downstream effects, okay? That downstream cascade allows for the expression of CR2. So you interact with the antigen at the follicular dendritic cell. That triggers the B cell to say, okay, there's antigen I recognize. Now it's going to activate and start really expressing the CD2 receptor. Now remember, CD2 interacts with C3d. Okay? That makes sense? What? Oh, yes, I'm sorry. I'm so sorry. CR2. Does that make sense?
Do they need to find the right pathogen, or is it any pathogen?
They do. They're B cell receptors. So we talk about all that B cell maturation.
Right.
This B cell receptor is specific to one epitope, really highly specific. So if a B cell comes in, moves to the dendritic cell, and finds a partner, it starts this activation cascade. Important to note, if that B cell doesn't interact with anything, it just keeps on its merry way. It loses the CXCL13 receptor and enters the lymph system again to move to the next lymph node. Make sense? Thank you for clarifying that. Yeah.
Sorry to ask a question that's a little bit off topic, but are the NOD and TLR receptors highly specific as well, or are those binding anything?
They're widely binding. So TLR receptors, I know less about the NOD, one, because they're not really talked about in the book, and two, I think we know a lot less about them. But TLR receptors bind to big classifications. So TLR4 binds to any lipopolysaccharide. TLR9 binds to any unmethylated DNA. Or any unmethylated CpG region, I apologize. So it's big classifications of molecules they can bind to. Of course. Any other questions about this initial step? Okay, from this step, now CR2 is expressed, and that CR2 can then interact, I'm sorry, this is small, can interact with C3d. Remember, complement has made the C3 molecule that then the B cell is essentially interacting with the CR2 receptor. Make sense? That CR2 receptor activates a further cascade of protein activation within the cell using the classical pathways like NF-kappa B. Okay, sound good? At this point, our T cell receptor, or our B cell, is not an effector B cell. Still is not fully activated. Instead, what happens is it re-expresses CCR7. Remember, CCR7 was made down here by the HEV cells. Does that make sense? So by re-expressing CCR7, the B cells now leave the follicular dendritic cell and move back towards that HEV. Remember, they aren't fully activated yet. They're not fully effector B cells, but they're moving back towards that HEV. Simultaneously, we started by talking about T cells. Simultaneously, activated T cells, once they're activated by that dendritic cell, move towards the germinal center. What happens is the B cell is coming away from the germinal center, the T cell is moving towards it, and there's this zone in which they interact. That interaction zone allows for B cells and T cells to look for one another to find a cognate pair. Okay? When that cognate pair is found, using MHC class 2, when that cognate pair is found, the T cell expresses CD40 ligand which activates the B cell by interacting with CD40. Make sense?
What is the cognate pair? What do they have to do?
Cognate pair is a term used for a B cell that expresses an antigen that a T cell recognizes. And it's the pair of a T cell and a B cell that are recognizing the same, or the T cell that recognizes an antigen that a B cell is presenting. Does that make sense? Yeah,
So
Nani.
at this point, they've encountered two antigens, one on the dendritic cell, and then you're saying one on the T cells.
Clarify they.
The B cells. B cells have encountered an antigen on the dendritic cell, which upregulated, it led to CR2 expression. And then now again, it's encountering another antigen shared with the cognate antigen in the T cell.
So the B cell is actually presenting the antigen. The B cell has the MHC class 2. Right? So the B cell gets activated at the flick of the dendritic cell, takes in that pathogen, dissolves it in an endosome, and then presents it on MHC class 2. That MHC class 2 then can be recognized by a T cell. Does that make sense? Important to note, again, that peptide that's being presented by MHC class 2, this is a common theme, doesn't have to be from the same pathogen that the B cell interacted with. Right? The B cell interacted with some epitope and then sucked in everything around that epitope, which means that anything it sucked in could be presented to a T cell to activate the T cell. This has importance when we make vaccines, because the B cell is presenting peptides that it takes up after interacting with its receptor. Does that make sense?
Does the B cell always have to present the antigen to the T cell?
Yes.
Okay, so this happens every single time for the B cell to be activated before it can start making antibodies. We didn't get there yet.
Right. Before it can, we'll get there, but before it gets fully activated, undergoes or goes back to a germinal center, does its thing, it must interact with the T cell. Activation without a T cell, these are these T-independent activators. I have a slide on it, but those are the only ones that don't require a T cell. The rest require that T cell interaction to get the robust antibody response. Yes?
And in terms of interacting and finding a T cell, is it any T cell that recognizes the MHC2, or is it like that specific antigen?
It's any T cell that recognizes the specific antigen that the B cell is presenting. And remember, at the review session this was mentioned, the B cell is going to present all sorts of antigens. So the B cell takes in, just like a phagocyte, takes in everything around that's around this receptor interaction, takes everything in, dissolves it up, and puts it on MHC. Which means that if you take a peptide, let's say a peptide that is 100 amino acids long, the B cell takes that in, it's going to divide that 100 amino acid long peptide into a bunch of little fragments. All of those fragments get loaded onto MHC class 2 and presented. And a T cell only needs to recognize one of them to get that cognate pair. Yeah.
So T cell receptors are very highly specific for the proteins that are being expressed by T cells.
Yes. Yeah.
That's not unclear on CR2 being expressed on the T cell. So I understand why it was expressed on the follicular dendritic cells, the whole pathogen nucleus. But why exactly do you need present like CR2 on the T cell?
So the CR2, it's a good question. The CR2 allows the B cell to recognize complement. So you don't, should clarify, you don't always have to have that complement interaction. B cell can be activated without that. But that complement interaction allows for a more robust response. So that CR2 is what allows the B cell to recognize it's got an antigen, and that antigen is bound to complement. So it ties together the innate response with the B cell response. Does that clarify?
So the classical pathway has to go through this lymph node interaction in order for the complement classical through B cell to pass that in. I don't know, that's my question.
So I think to clarify, the alternative pathway and mannose binding lectin pathway happens before that complement can be attached to the antigen that the B cell is seeing. Which is why there's that big time delay, part of why there's a big time delay, right? That complement system has to put on the complement protein. That protein then has to get all the way to a lymph node where a B cell can recognize it. And then the B cell still has to get activated, right? But it's a good question. Any other questions before we continue? Okay. So we have a cognate pair. That cognate pair is composed of a T cell that recognizes one of the many peptides that a B cell is presenting. Okay? At that point, the interaction between CD40 and CD40 ligand causes the B cell to re-express the CXCL13 receptor. So that's CCR5. I don't think we need to know all the protein names, but know that once that cognate pair has interacted, they go back to the germinal center. This is where the process of somatic hypermutation and class switching both take place, in the germinal center. And that's after the B cell has interacted with the T cell. Make sense? So they go back to the germinal center. The B cell undergoes somatic hypermutation. I can't remember if I put that in here or not. Undergoes somatic hypermutation in which the same aid protein that was used earlier for class switching, we talked about it, makes little nicks in the DNA and induces random mutations. Those random mutations allow the B cell to produce different variations of its receptor. As it does that, it then interacts with that follicular dendritic cell. Remember, that follicular dendritic cell is expressing all of the pathogen antigen, right? So that B cell is mutating all of its receptors. It then goes back to the dendritic cell and checks itself. If the receptor still binds to an antigen, that B cell is kept alive and then can go either to a memory cell or a plasma cell. However, that random mutation can just as likely result in an antibody that doesn't work, right? So if it doesn't work, that B cell doesn't connect with the follicular dendritic cell and will go through apoptosis. So we lose a ton of B cells in this activation pathway because random chance says that you're probably not going to get one that's more selective. But this also allows for the antibodies to mature and become really, really selective, which is important when we're dealing with toxins. You want your antibodies to be super selective to whatever pathogen you're fighting. And this somatic hypermutation is how we do that. And I should also note, the point of a T follicular helper cell in this whole interaction, the reason you need that cognate pair is that helper cell is going to give the survival signals to the B cells. So if the B cell interacts with the dendritic cell, it will then cause that T cell to give its survival signal to allow the B cell to continue in its maturation pathway. If it doesn't interact with the dendritic cell after the hypermutation, it doesn't get the survival from its T cell partner, and it dies. Make sense? Yeah.
So somatic hypermutation happens after the B cell's already been presented with the antigen?
Yes, because the B cell must be activated to undergo somatic hypermutation. It still isn't an effector cell yet, but it's an activated cell. Yeah.
So when the B cells are coming out to like that zone, right, when the cognate pair happens, if they don't bind, right, and they undergo apoptosis, do they have to like leave that zone, or do they just die in the zone?
That's a really good question, and I'm not 100% sure. I'm fairly sure there's phagocytes in that whole B cell region that clear out all the dying cells. And presumably the same reason that all the non-activated cells get pushed out, all the activated cells are heading towards the dendritic cell and push out all of that pathogen. Yeah, pardon.
Is the T follicular helper cell like a specific T cell type, or is that just a function served by T cells that migrate them?
It's a specific cell type. Of those 6 T cells, one of the types is a follicular helper cell. And their role is to sit in the lymph node and help B cells to get activated. Yeah.
Is the IgM response happening at this point because it doesn't have to go through somatic hypermutation?
The IgM response?
Yeah.
It can. So at this step, before the cognate pair moves back to the germinal center, some of those cells could just remove themselves from a follicular helper cell and start secreting IgM. And at that point, they would become an effector cell. But a majority of them will head towards the germinal center to continue somatic hypermutation. Clear as mud? Okay. All right. It is at this time that they undergo class switching. We talked about class switching, we talked about antibodies. This is what allows a B cell to produce IgA, IgE, and IgG rather than IgM and IgD.
Is there a specific reason other than the fact that it's like after IgM that IgD also gets made with IgM? Or is it because they're the first two?
Primarily because of the first two, but also because there's splice sites. So we learned about the spliceosome way back when, last December or whatever. There's splice sites between IgM and IgD, so that when that RNA gets kicked out, it can splice to either IgM or IgD. Within the textbook, as of note, IgD, we really have very little idea what it does. It's a very kind of ambiguous, we don't know why it's there. So IgM would be the primary default that gets actually displayed and then presented. Any other questions? Okay. So, class switching. After the B cell has gone through its somatic hypermutation and it's ready to go, it's got a receptor that's really highly specific, it then interacts again with that T follicular helper cell. The cytokines that are made at that point are what determine which one of the classes that antibody will be. So it can then, just based off of its interaction with the follicular helper cell, gets determined or gets directed into which type of the constant region they're going to express on their antibody. Does that make sense? So it all just comes down to that cytokine interaction with the T helper cell. When that interaction takes place, there's an additional step in which that B cell can become a plasma cell as an effector cell, or it can become a memory cell. This is based, as the example we were given in class, if IL-10 is present, it becomes a plasma cell. However, if IL-4 is present, it becomes a memory cell. So this, again, happens downstream of all that somatic hypermutation. It's kind of the last step is, okay, I'm now activated. I've got an antibody that's specific. I know what class I am. What do you want to do with me? Do you want me to be a plasma cell, or do you want me to be a memory cell? And from that point, they then leave the lymph node and enter the circulation. Any questions at all about B cell development before we move on to the next step?
I thought IL-10 was anti-inflammatory.
Yeah.
It's promoting inflammatories.
Yes. In this instance, it is promoting the production of the plasma cells. Part of the problem of categorizing everything as either one or another when our body uses things for everything. But in this case, IL-10 is going to result in plasma cells. All right, let's talk a little bit about the function of antibodies, because this is where it comes all the way full circle. Once we have an antibody that's made, specifically IgG, that antibody can then bind to a pathogen. When it does, we then can activate the classical complement pathway. This uses the IgG as a binding site for C1. It's essentially the equivalent of the mannose binding pathway, but it uses antibodies instead of the mannose binding lectin. Okay? And this can work with IgG or IgM. The important part is that it has to happen after the adaptive immune response. So we start with complement, and we circle all the way back around to complement by using this IgG. Additionally, IgG, many phagocytes have an Fc receptor on them, and if IgG is attached to a pathogen, the phagocyte will get a hold of it and phagocytose it. So the whole point of getting the adaptive immune response is to create antibodies that signal complement and signal phagocytes, and then work on opsonization and detoxification. Does that make sense? And the other arm would be the cytotoxic T cells, which just go directly and kill any sort of infected cell. All right. I want to touch upon complement clearance, because this was mentioned in class. Complement clearance is what happens when you have a bunch of antigen. All that antigen can be attached to an antibody. With that antibody, complement can come in and put a C3b molecule. So here you have antigen attached to antibody, which allows the C3b to be deposited. That then interacts with CR1 on blood cells. The blood cells then go to the spleen, where macrophages in the spleen pull off the, move this out of the way. Macrophages in the spleen pull off the antigen and get rid of it. So this is how we get complexes of antigen and antibodies cleared from our system. If we didn't have this, antibodies would keep building upon themselves and we'd get deposits. This actually is an autoimmune disease I don't think we've touched on yet, but I'm sure we will. But this is a really important pathway for getting rid and clearing complement. All right, so let's quickly summarize, put it all together, and then I have just a couple last ending thoughts on some of the other things we've touched on. So if we put it all together, a pathogen enters the skin. When it enters the skin, it's recognized by phagocytes and complement. Complement and phagocytes then lead to the inflammatory pathway, which recruits neutrophils to come in and try and phagocytose and get rid of the pathogen. If that doesn't work, then dendritic cells migrate to the lymph node, where in the lymph node we have B cells and T cells coming in all the time and scanning dendritic cells and follicular dendritic cells. If an antigen is present because it hasn't been eliminated by the innate response, then we activate our adaptive response. And our adaptive response starts making antibodies, starts making T helper cells, cytotoxic T cells, in an attempt to increase the amount of complement, to increase the amount of antibodies that can come in and protect our cells from all these pathogens, and to try and eliminate whatever's infecting us. And then at the end, we have CTLA4 and other cytokines that cause a resolving of the immune system. We haven't touched on it yet in class, so I'm not going to get into it now. But at the end, just know that we have to turn the immune system off. We can't have it hyperactive all the time. Any questions on the entire cascade? Conceptually, it starts with complement, and I think of it as a nice big pathway that ends with complement and antibodies. And if we put it all together, this happens on a time scale where initially, after 1 day, you can start to get the innate response, where you've got neutrophils invading, you've got macrophages that are destroying pathogen, and then in about 4 days, you start the adaptive response, and it maxes out in its capability at about 10 days, or 12 days, 10 to 12. Does that make sense? Okay, so that takes care of the timeline. Vaccine development. This was hit on a bunch, so I want to make sure we have this nailed down. Vaccines utilize,
Wait, sorry. The C3d that is formed, is that only from the alternate pathway? Or is that from alternate and
The C3d comes from any time that you have a C3b that binds to CR1 and protein I can come in and cleave it. You can get C3b from the alternative pathway, from the MANNOSE binding pathway, and from the classical pathway. So that C3d formation comes from any of the complement pathways. Does that make sense?
But technically, like, the classical one would start after the B cells are activated.
Yes, the classical would start after the B cells are activated because you need that antibody, right? There's also C-reactive protein that adds to the classical pathway, and that comes out slightly before antibodies. You can get that pathway activated, but that takes time to make C-reactive protein in the liver. But of note, the antibodies are produced for that third leg of the classical pathway. Any other questions? All right. Let's talk a little bit about vaccines. Vaccines utilize this activation of B cells and T cells in order to essentially induce the immune system to make memory cells. That's what a vaccine is for. It's to make memory cells so that if you get infected by it, you've got a rapid immune response that can clear it. Okay? Importantly, T cells cannot see anything but peptides. So T cells have to interact with peptides, whereas B cells can interact with pretty much any epitope. And if we have a goal, if we walk through, the way I think about this, I kind of think about it as the process for making a vaccine. So if we start, we want to make a vaccine to a polysaccharide. This is something that's presented on a bacteria we want to be vaccinated against. To do this, we need to make that tiny little polysaccharide presented to B cells. That's pretty easy. A B cell usually will recognize any sort of foreign molecule. We've got such variability. However, we have a problem because B cells need help from T cells. They won't go into the highly activated state unless they interact with a T cell. But if you're trying to make a polysaccharide antibody, there's nothing there for a T cell to interact with. So our first solution is to add a toxoid. A toxoid is a small bit of peptide that we attach to whatever antibody we're trying to make, or whatever antigen we're trying to make an antibody against. Does that make sense? So you attach a protein fragment to that polysaccharide. The B cell recognizes the polysaccharide, takes it in, and then it's going to chop it up and put all those proteins that you've attached onto an MHC class II receptor. That MHC class II will then present it. And now all you need is a T cell that interacts with that peptide fragment. So this is why it's so important, as I mentioned, that the T cell and the B cell don't have to be recognizing necessarily the same exact bacteria or the same exact thing. That peptide fragment could be anything that activates a T cell. You just want to attach it there so that you've got a B cell, so that you've got an MHC molecule that has a peptide in it, so that the T cell can recognize a peptide. Notably, you'd want to choose a peptide that is a foreign peptide so that you have T cells that are there. But any peptide that's foreign can be attached to that polysaccharide so that the B cell has a T cell that it can pair with. Once that T cell's paired, it then can go through its somatic hypermutation and make the antibodies we would like it to make. However, we've still got one more problem, and that problem is we need some way to activate the T cell, right? A T cell can't interact with the B cell until it's activated. So to do that, we use adjuvants. Adjuvants are small little bits of inflammatory creating protein that allow for we trace all the way back, allow for the antigen presenting cell to be activated. Because you need that antigen presenting cell to give B7 to the T cell so the T cell can get activated. So if you think about it from top to bottom of what do you want to do? You want to make an antibody. What do you need to get that done? You start thinking about everything that's involved, you can realize why a vaccine includes the polysaccharide that you want your antibody against, a peptide, in this case it's called a toxoid, and an adjuvant, because all three are necessary in order to activate the immune system. Yeah?
How do you make sure that the B cell is making antibodies to the polysaccharide and not the toxoid?
I don't know the answer to that. I'm not sure. Presumably there's no way to make sure.
I think B cells express when one B cell only expresses one kind of antibody. So if it happens to pick up that polysaccharide,
Right, but you could have another B cell that picks up the toxoid.
oh, okay.
But if you used, for example, a toxoid that is within that bacteria, it wouldn't be harmful. I think that's the idea of it. But that's a really good question.
How is a toxoid different from an adjuvant?
So an adjuvant is aimed at activating an antigen-presenting cell because it's got to get that B7 present, whereas a toxoid is aimed at activating the T cell. Right? Does that make sense? All right. So we have a special class of B cells. These are T-independent B cells. We've got two classes, Ti1 and Ti2. Main point here is they're much less efficacious. So if you have a T-independent antigen, it'll respond to that lipopolysaccharide, but it can't do class switching, it can't do hypermutation, and it's stuck with IgM. Likewise, a B cell that's responsive to TLR9 is very limited in its class switching and limited in its ability to proliferate. So know that these are there because we do have mechanisms for generating antibodies against something that doesn't have a protein, but our mechanisms are very limited. Additionally, children don't express these until they are 2 years old, and they're not maximally expressed until they're 5 years old. So there's a time period where we have a gap in our immune system. Yeah, make sense? Okay. I just wanted to include here, we talked about this, but for anybody that has the PowerPoint, I included the specificity of the antibodies. IgM is a pentamer, flows in the circulation usually. IgD, we know very little about it other than it's on a B cell, it's part of the B cell receptor. IgA, as a dimer, this is what's secreted in all mucosal immunity. Of note, mucosal immunity lacks inflammatory proteins, so just if it's in the mucosa, it won't have inflammation. IgE is actually a co-receptor for the eosinophil and is really important with allergies. So we don't have problems with parasites very often, but we have the residual IgE, which causes a lot of problems with allergies. So allergies, IgE. And then IgG is the most specific antibody we have. It's the most dispersed within our body, and it's very involved in the complement activating system. So IgG is really important to get that final step. I included this table for anyone who's interested in it. Just shows where they're all used and how they all get applied. Briefly, going to mention checkpoint inhibitors because those were also talked about in like 3 classes. A checkpoint inhibitor is designed to, as it says, prevent a checkpoint. So to put this in context, cancer cells use both CTLA4 and PD-1. Remember, CTLA4 suppresses the immune system, and PD-1 actually causes the T cells to commit apoptosis. So a cancer cell avoids the immune system by presenting CTLA4 and PD-1. So if a T cell does interact, it gets killed or it becomes anergic, right? So the way we get around this, or the way that treatments are getting around this, is by making a checkpoint inhibitor. If you block that CTLA4 on the cancer cell, or if you block its PD-1 receptor, it can't prevent that T cell from activating. Make sense? So the goal of a checkpoint inhibitor is to prevent the abnormal suppression of our immune system. As part of this, checkpoint inhibitors often have side effects of hyperinflammation because our cells no longer have the capability of suppressing the immune system. Does that make sense? Cool. All right, a little bit on mucosal immunity. IgA dimers are used. There are no inflammatory receptors, so you don't get this big inflammatory response. This is important because the epithelial cell on the gut is the only block between the commensal bacteria and our cells. So you don't want inflammation because then it can open a big hole that all of your commensal bacteria can get into. So if you suppress some inflammation, you usually hold that endothelial line intact. We have intraepithelial lymphocytes, which are actually activated effector cells just sitting in the epithelium. The reason these are there is because if some sort of pathogen gets through the mucus, you want something there to kill it right away. So we actually have activated cells there. And then the M cell transports, it's just like a monitoring cell that transports all sorts of antigen from the intestine into the lymph organs that are right below the intestinal epithelium. And then I think we're down to just a couple last slides. Thank you all for bearing with me. The last couple things I want to discuss are immune evasion. Pathogens have developed, just like we have, in parallel to evade every step that we make, and we try and fight every evasion that they have. One of these is a variable surface glycoprotein. So the adaptive or immune response takes time to produce antibodies. If the pathogen is always changing what it expresses on its surface, by the time we have antibodies made for it, it's no longer using that surface protein and it's no good. So a lot of pathogens have the ability to change their surface proteins so they can always evade our immune system. And then additionally, A lot of the viruses make inhibitory proteins to MHC. I talked about this when we were talking about how MHC is loaded. Many, many viruses either cause the degradation of our native MHC molecules or prevent the protein peptide fragments from getting loaded into the MHC. And in that way, if the peptide is never loaded and the MHC is not there, there's no way for our immune system to detect it. This is a point that was brought up, this version of immune evasion was brought up when discussing the process by which memory cells are activated and the primary activation pathway is used. So once a memory cell is activated, it is the default cell for that same exact pathogen epitope. So if you have memory bodies in your system, or memory cells in your system that make antibodies, for all of these epitopes, anytime that pathogen enters the system again, it's going to use the memory cells. However, over time, pathogens mutate. And so what happens is with time, if they mutate once, now you still have memory cells, but now this one is different, and you're now lacking one of your memory cell subtypes. It continues to mutate until you get only this final A epitope that your body recognizes. So your immune system becomes slightly less efficacious as pathogens mutate because you've got less memory cells that are effective against them. Additionally, when the memory system is used, the innate or the adaptive system that would generate no more antibodies is suppressed. So you won't make additional epitopes for any of these if you've got a memory system that is intact. What this means is that over time, you lose the ability to react to the pathogen because you have less effective memory cells until eventually all of the epitopes are new and you have to go through your primary response again. Does that make sense?
Is that why you need boosters?
That is why you need boosters. Additionally, it's very unclear as to why this happens, but certain antibodies and antigens last longer than others. So some vaccines we get, we never have to get a booster for, because for whatever reason our immune system keeps around forever. Other ones we always need to get boosters. This is, I would say, more the reason that we have to always get influenza vaccines or something that's always mutating because our memory response gets worse and worse with time. Yeah, Karen.
So it's easy to think about in the term of like first infection and then second mutation, or first mutation, second mutation, all of that. Why does that correlate exactly with the second infection and third infection? You know what I mean? Like, does it only
It doesn't.
mutate? Oh.
It doesn't. They're just using that as a way to delineate these columns. But that pathogen, if it's the same the first 5 times you're affected, your immune response works great. It should be like first mutation, second mutation, third mutation. All right. Hopefully this helped to put a little bit of perspective on the immune system and didn't just confuse a bunch of people because I'd feel really bad. I want to say thank you for coming. I do have practice questions that I have in here. If people want to stay and do them, I'm happy to stay and work through them with people. But I understand that it's been an hour and 45 minutes and you're all probably chomping at the bit to get out of here. So I'm happy to take any general questions. Otherwise, thank you all for coming, and I really do hope this was helpful. I'll be sending out a survey afterwards. I'd appreciate any feedback you have for me. Thanks, everyone.
Yeah, so you said that when you were talking about vaccines,
when we were talking about vaccine development, I think you had said that adjuvant allows the toxoid allows the T cells to be activated, and the adjuvant allows some. I can't remember exactly, but is it switched, or is
so
that?
The adjuvant allows the antigen presenting. The adjuvant is an antigen presenting cell.
Right, that's right.
The toxoid is important to get that interaction between the B cell and the T cell.
Right. Oh, okay.
So it's that like final step.
Right.
Of that.
It's needed to activate the B cell and allow it to interact with the T cell? Or do you not need it even to activate the T cell?
You need it to get that final interaction with the T cell, which is required for activation.
Right. And the adjuvant is activating APCs, which allow the T cells to be activated. Okay. Thank you. Thanks for doing this. It was extremely helpful. So thank you.
Are the people that are still sitting here wanting to go through practice questions? Okay.
Where do you find like the answers to these questions?
Oh, they're in the book.
From the same book that you told us from the beginning.
Yes.
Thank you so much. Thank you so much for putting this together.
Yeah.
Super.
I hope it was. Okay. I'm glad. All right. I have put the first question up. It is a hefty question, so I'll let you have time to read it, and then we'll talk about it. Additionally, I'm just going to mention, for these types of things, I'm considering making little recording videos that are like little snippets of like just B cell activation and just T cell activation, or whatever it happens to be for future class sections, and just sending those out instead of doing big lecture. So if that way more people can watch them on their own time if they want to. Anyway, I'm considering doing that. I'm going to try it with this one, and if it works, I'll probably start doing that in the future, just so you all know.
We were talking about doing this and like going through and making the heavy chain and making the light chain, and then you have like 3 or 4 chances. So what, and it's like recombination, but it made it sound like was that like this peptide's already created and then it's manipulating that. You understand what I'm saying? So when you're making the heavy chain or light chain, you talk about having 3 chances to come up with something that's gonna work. Right? So how does that happen if we've already expressed this?
Okay, so what happens is you express the B chain, keep getting T cells. You express the heavy chain.
Yeah.
The heavy chain then pairs with the surrogate light chain. The surrogate light chain.
Which could bind to any heavy chain theoretically. It's like a universal light chain.
Yes, it's universal.
Okay.
Because all this is so you can get through checkpoint 1. You then make a light chain. Okay. Once you make the light chain, you then go to selection. In selection, if it's found to be too tightly binding, it's still on a stromal cell. And so it still has all of the signals it needs to then remake the light chain. So if I'm just going to go to the next
I see.
slide.
So it like destroys that peptide that it created
Exactly.
and makes a new peptide.
Because you've got 4 regions on your genome that code for different light chains.
Oh, okay.
Right? So if your first K chain doesn't work, you just go to the next one.
Okay.
Go to the next one. And it does this in the B cell specifically, if I remember correctly, 1 time. So if it doesn't pass negative selection, it can send it off for light chain recombination once. And then if it doesn't work, it's like you're too far gone.
Okay.
And part of this just happens to do with you start cutting out more and more of the DNA every time.
Okay. So where is this taking place? Because it's not. This isn't being expressed yet on the membrane.
This is all being expressed on the membrane.
It is.
And it's all happening with the stromal cell. So in the very first one, I show it linked on a stromal cell. All of this is happening while it's linked onto a stromal cell. And it's all being expressed because the expression is what allows, being expressed like this, if it expresses that T cell receptor, which is this little tiny Y, that's what allows other cells to come in and check it. So for selection, another cell will come in, check it, pop off. This interaction here is what allows it to check to make sure you've made a functional heavy chain. It'll roll over, and if it makes a functional heavy chain, it'll bind to its adapter light chain and its surrogate light chain with its heavy chain, right? It'll pass selection, or pass checkpoint 1, and then it'll make a light chain.
Okay. That makes sense. Thank you.
Yeah, of course. All right. Is there any brave soul who wishes to throw out an answer for which this is? Yeah, Kelvin. It is. Why?
But it seems like all the antibody levels are low, so I'm
Mhm.
you have the normal T cells, which will hyper IgMs. So XLA is your gluton, I think. Gluton something deficiency, which leads to the capability to like deal with your like changing the B cells.
Right.
Due to deficiency. I can explain that better.
So essentially what's important here is, yes, the antibody levels are very low. To make an antibody, you require Bruton's tyrosine kinase. And this is important in the, I gotta remember, I think it's light chain part. I would need to check. Hold on, I just want to make sure. Yeah, BTK is a kinase that's used in the developmental process of a B cell. If that kinase is mutated, it can't generate antibodies because it won't ever make a functional antibody, so you get these really low levels. All right. Yeah, let's go to the next one.
Sorry, was the answer B or D?
B, I'm sorry. B is the answer. X-linked A gamma globulinemia. For the purpose of this, DiGeorge. Does anyone remember what DiGeorge involves? So DiGeorge, yes, it's catch-22. And the important part about DiGeorge is that you get thymic involution. You just don't get a thymus that develops. So with that disease, you don't get any T cells. And so all you're able to make with DiGeorge would be your T cell independent antibodies. So some IgM, maybe a little bit of class switching from the TLR9 receptors, but your antibody repertoire is very limited without T cells. All right. Second question. Also, feel free to discuss with people if you'd like. All right, anyone want to shout out an answer? All right, is it A? Why not? Okay. Is it B? Why not? Is it C?
Why would it not be?
So there is a small amount of negative selection that can happen in secondary lymphoid tissue because you're getting a lot of interaction with T cells and B cells and MHC receptors. There is the potential for autoreactive signaling to take place. I tried reading about it. It's really vague in the book. So I think B is like a really tricky answer because we learn it happens in the primary lymphoid tissue, but technically negative selection can happen in the secondary tissue. What about C? Why not? Yep. And D? Okay, so E, right? Negative selection, no matter whether it's B cells or T cells, is always getting rid of auto immune cells. All right, 7-4. Is it C? Let's try a different one. Is it B? Why?
T-cells are always MHC.
Not quite. T cells are always obligate MHCs, right?
Right.
So, oh, did you say dependent?
Yeah.
Oh, sorry, I heard independent. Okay. Yep, we're good. How about D? Why?
Don't you not need your thymus as an adult?
Yeah, so the operative word in this question is actually adult. Because adults don't need their thymus. In fact, most people don't have an operational thymus. So in this case, the answer is E, where you don't need any sort of thymus, because once your T cells are made, as you're developing, that's what you have. That's them. Interesting research, just as a side. This is like the theory of why they're thinking that such a sterile environment that we have now is causing more allergies, because our T cells are not being used because we don't have any sort of exposure to pathogen when they're developing in the T cell in the thymus. Anyway, just as a side, that's part of the theory of why we think we're getting more allergic to things. All right, 7-9. And I do apologize for the fuzzy images. All of these were pictures that I took.
A question
Yeah.
about George.
Mhm.
So that's just more prevalent in children then, or when their T cells are still developing?
Yeah, so it's a deletion of the Q11 part of the chromosome 22.
Oh, okay.
So if you have it deleted, it's just a congenital disease where you never develop the thymus. All right, anyone want to propose an answer for this one?
C. I mean, I thought B. I thought that was like too strong.
What answer did you propose? I'm sorry, it's really hard for me to hear up here.
Isn't it after positive selection you change from double positive to single positive?
So positive selection is where your T cell is going to interact with an MHC,
Yeah.
and that's where it's going to decide what fate it's going to go.
Yes. So then after that, would it become single positive?
Yes.
So then it would be B.
Yes, the answer is B. All right, 7-10. This is, again, I'll just let you know, this is an operative word. So I think there's several answers that are like, yeah, it's kind of true. There's one that's like definitely the best. Anybody want to propose an answer? Why would you propose C?
Because in order for T cells to recognize an antigen, the antigen has to be on an MHC molecule.
Right. So this is, again, as I tried to iterate through the review, that T cells require a peptide and require that peptide to be bound to MHC. So when you say MHC restriction, it means that they only will interact with a peptide that's bound to an MHC molecule, which would be C. to take a ponder at the answer? Why would you say E?
Because if it's double positive, that means it hasn't yet gone through positive selection. And so if it binds to the self MHC class 1, that would dictate it towards CD8.
But if it's not going to be
What?
Oh, low affinity is, oh,
a low affinity.
I was thinking because it's a self-peptide.
Are we thinking A
Should be low affinity.
for the self?
Yeah. I was thinking A because it shouldn't be binding to itself, but it could be exposed.
But the first selection is only does it bind to MHC, not necessarily to the content.
Right, so there's kind of two important aspects to this. One, you're correct that it's a self-peptide, right? Two, it's MHC class 1, but the operative word is the low affinity, okay? In the thymus, if you bind to self with low affinity, that's fine. So it's kind of a red herring, this idea that the self protein, right? The important part is that it's low affinity and that it's MHC class 1. So you actually get positive selection for that CD8 cell.
So if that's at high affinity, it would definitely be A.
Exactly. Because this would be testing whether or not it interacts with MHC more than that self-peptide. Right, like this is part of positive selection, where it's testing what MHC class it interacts with and making sure it interacts with MHC. So it passes the test, it interacts with MHC, but it's low affinity, so it stays living.
And what if we were already in single positives, we were already expressing that? Would that also make a difference if we were in negative selection and it was binding at all?
No. You can get, in fact, the book even describes it as moderate binding. You can get, there's a threshold amount at which you can bind self-antigen, which again is important in autoimmunity, right? We have so many autoimmune diseases that are all a result of things escaping the thymus. All right, anyone want to answer A? Okay, antigen presenting cells. What are the antigen presenting cells? Yep, so B cells, dendritic cells, macrophages. Those are the big ones. Let's see. So yeah, which of these populate the thymus and play a role in selection? I think actually for the purposes of this exam, I think that is very low yield to put it because they don't really seem to care what cells do it, if that makes sense. For your knowledge, the thymus is filled with macrophages and dendritic cells, along with thymic epithelial cells, which are a special lineage that only exists in the thymus. And the thymic epithelial cells are what primarily do the positive selection, whereas the macrophages clear out the cells after negative selection.
Is that the same as cells?
If you're talking about those that are in the thymus, then yes. Usually they're described as a cortical epithelial cell or a medullary epithelial cell in the thymus. And follicular usually is used with dendritic cell in the lymphoid. Okay, yeah, cortical epithelial cells are a part of that lineage. Oh, sorry. Okay, this is actually, I think, a good question. Does anyone have a idea on C?
Did we answer like which cells mediate positive and negative? Is it for epithelial positive?
Oh, I just want to make sure I say it correctly. So the cortical epithelial cells mediate positive selection and somewhat negative selection, and the macrophages are more responsible for clearing the debris. And then just to add more layers to it, the macrophages and dendritic cells play a role in negative selection primarily. And does anyone have any idea about C? Yeah, Carolyn.
So if you're only presenting or only using the thymic environment to educate the T cells, then those are going to be the kinds of T cells that you have in your body that you want them to be taking in things from outside. I literally made that in the
B cells that are just self-reactive.
For C. Yeah, so the idea is that if they're in tissues, they could soak up self-peptides and then present it and you'd get autoreactivity. So yes, you're correct. Additionally, if they're in the tissues, they're not being surveyed by the lymphocytes or the lymph nodes, so there's less exposure to them.
And that's because that's an exogenous thing that's occurring, whereas with that HC1, it's endogenous.
Exactly. That's
You'd be taking in things from the outside versus
exactly.
taking things that might get hurt inside.
Yep. So this kind of recaps a little bit of the B cell activation pathway. Does anyone want to explain how B cell becomes essentially trapped in a lymph node at the boundary between B and T cells?
It needs to find its cognate partner in the T cell, otherwise it'll just hang out there or leave to go to another lymph node.
Exactly. And why would this be necessary?
Just like another step of regulation.
So what happens if it doesn't get stopped at that border zone and it leaves? Yes, but more importantly, the T cell is what gives it the full activation potential. Once it leaves, if it were to leave and not get stuck there, it wouldn't have the ability to interact with that T cell and get fully activated. Neither would it have the ability to go back to the dendritic cell and undergo hypermutation and class switching. Bless you, bless you. All right. Anyone have any ideas? Calvin.
Answer E.
Why? So he asked if the answer was E.
So you need the T cells from the thymus to activate class switching.
Right. Again, taking it back to pathway, you need the T cells to activate the B cells so that they can undergo class switching. All right, anyone want to throw out a guess for this one?
I know it's IgA, but I'm not sure.
It's dimeric.
It's like with the JJ like
Exactly. And the reason that's important, we didn't so much get into it in class, but the J chain allows for the transport through the epithelial cell. So if you don't have it dimerized, it doesn't allow for that transport through that system. Why?
They have like the CR1 receptor which picks up C3b.
Exactly.
Is that right?
That's exactly right. So yeah, the red blood cells contain CR1. CR1 is a receptor for C3b. And together, that allows the erythrocyte to take it to the spleen for cleaning, essentially. I think this is the last question.
How do you read things like this? You know what I mean?
One
Like,
word
you
at a
start
time.
to talk to them.
Sorry, that was really sarcastic.
What do you think?
I said one word at a time.
But like on tests,
You're supposed to read the last slide, and then if you can't figure it out from that, then you go and look at the test like numbers, like any numbers they give you, and then you read the whole question. You're still gonna get it.
Yeah, I'm a bad person to ask test advice for, because I just read the whole question.
I do too. That's why I take time.
Well, yeah, it's like a good problem.
All right. Any takers want to throw out a guess? D? Why would it be D?
Drag next to the mix, or like decides like what gets sliced out, and digest the first one, we're just gonna do some more of that.
Okay, what step in development is REG used? What step in the antibody development is REG used? What?
The gene
Not somatic hypermutation. It's somatic recombination. So REG is used. Remember, in the gene, you've got like 30 J segments, 30 V segments, 30 D segments. REG is used to combine those, right? So it doesn't matter at that point whether you are IgA or IgM or IgD. REG is used upstream of that. You can't make an antibody or a B cell receptor without REG because you can't isolate just one V, D, and J segment. So it can't be REG1 because then you wouldn't get any antibody. Okay, so now we've got rid of REG1.
Is it C3 or something that's T cell related so that
Okay.
B cells can't interact and then go through this whole
So would it be A because that's needed to activate the B cells?
Exactly. This is hyper IgM syndrome, is what this is describing. And in this case, you're lacking the CD3 or CD40 ligand, which is necessitated in order to activate the B cell in the lymph node. So if you lack that ligand, all you can do is make IgM. You can't make any of the other subtypes. So they talk about being severely depressed in IgG and IgA, and that's because without that CD40 ligand, you can't fully activate your B cell and will never undergo class switching.
And that CD3 ligand
It's on a T cell. And then CD3, I just want to check before I say this out loud. Yeah, so without CD3, remember that is the co-receptor for the T cell receptor. So you've got a T cell receptor, and on the sides is CD3. Without it, the T cell can't even develop because it can't go through its selective process in the thymus. So that explains CD3. And then CD19 and CD81, this is part of the CR2 complex. We don't need to know those proteins, so that, regardless, it's only part of that minor part of the B cell activation pathway.
Could this also be caused by a defect or deficiency in AID, or like that thing that like puts out IgM or IgE?
I would say yes, but I would need to check that. Like, it logically follows that if you lose AID, you also lose the ability to class switch.
Yeah.
Though I don't know if it presents in exactly the same way.
But, okay, yeah. It's
Yep.
just like a mechanism.
Yeah, I would say yes.
So for CD3, if that would also be, it would be the same thing where you would make IgM either.
Exactly, because your T cell needs that co-receptor in order to go through positive selection.
So the big thing is that IgM. It's
Exactly.
Yeah, okay.
So if it's hyper IgM syndrome, it's going to be class 40 ligand usually. All right. Thanks, everyone.